So our next topic is double fertilization. The complete pollen pistil interaction. Pollen pistil interaction means all the events from pollen deposition on stigma until the entry of pollen tube into the embryo sac of ovule. Filiform apparatus guide the pollen tube into the embryo sac. Okay. Then finally 2 male gametes are discharged into the cytoplasm of synergids. Finally 2 male gametes discharged into the cytoplasm of synergids. And 1 male gametes move towards egg nucleus and another male gametes move towards central cell or polar nuclei. Okay. We know the structure of embryo sac. This portion is called chalaza and this portion is called micropylar region. Within the chalaza, 3 antipodal cells are moved towards the chalaza. Three antipodals move towards chalaza. Then polar nuclei located at the central portion. That cell is called the central cell. Okay. Then 1 egg cell and 2 synergids move towards micropylar region. Okay. Then you can see here filiform apparatus. Okay. This is antipodals and this one polar nuclei. Okay, and these cells are called egg cell and this one are called synergids. 3 antipodal, 2 polar nuclei, 1 egg cell and 2 synergids. Totally 8 nuclear structures are formed. Okay, then what happened? Two male gametes are discharged and 1 male gamete move towards polar nuclei and another male gamete move towards egg cell. Okay, these male gametes are discharged primarily at the cytoplasm of synergids and it move towards 1 male gamete move towards egg nucleus and another male gamete move towards polar nuclei or central cell. Okay, then what happened? 1 male gamete fuse with egg nucleus or egg cell and forming zygote. Male gametes are haploid, female gamete also haploid. So n plus n, 2n, diploid zygote is formed. Diploid zygote is formed. 2 male gametes are discharged. 1 male gamete fuses with female gamete egg. Haploid male gamete fuses with haploid egg cell and forming diploid zygote. And this process is called zygamy. Zygamy. Total 2 male gametes are discharged into the cytoplasm of synergids. 1 male gamete move toward egg nucleus and another male gamete move towards polar nuclei or central cell. Polar nuclei otherwise called diploid secondary nucleus. So 1 male gamete, haploid male gamete fused with haploid egg female gamete and forming diploid zygote. That process is called zygamy. That process is called zygamy. Okay. Then remaining male gamete. Remaining male gamete also haploid fused with polar nuclei. Polar nuclei is diploid. It is otherwise called diploid secondary nucleus. Diploid secondary nucleus. Total 2 male gametes are discharged. 1 male gamete, haploid male gamete fused with haploid female gamete egg and forming diploid zygote. That process is called zygamy. Remaining male gamete, haploid male gamete fused with polar nuclei. Polar nuclei otherwise called diploid secondary nucleus. Okay. And form triploid primary endosperm nucleus. It is simply we can say TPEN. It is simply we can say TPEN. TPEN. Okay. Triploid primary endosperm nucleus. Triploid primary endosperm nucleus. Okay, simply we can say TPEN. T means triploid, P primary, E endosperm, N nucleus, TPEN. Simply we can say PEN, PEN or TPEN. Triploid primary endosperm nucleus. And this process is called triple fusion. What fusion? Triple fusion. Triple fusion. So what happened? Totally 2 male gametes are discharged. 1 male gamete, haploid male gamete fused with haploid egg and forming diploid zygote. That process is called zygamy. Then what do you mean by zygamy? Haploid male gamete fused with haploid egg and forming diploid zygote. That process is called zygamy. Remaining male gamete, haploid male gamete fused with polar nuclei. Polar nuclei otherwise called diploid secondary nucleus and forming triploid primary endosperm nucleus. Okay. Remaining 1 male gamete fused with polar nuclei or diploid secondary nucleus and forming triploid primary endosperm nucleus. What is a ploidy? What is a ploidy? Ploidy is 3N, triploid. That's why it is called a triploid primary endosperm nucleus. How this triploid will be formed? N plus 2N. That is 2N. 2N plus N means 3N. 3N means triploid. Haploid male gamete fused with diploid polar nuclei. 2N plus 1N. That means 3N. Triploid primary endosperm nucleus is formed. TPEN or PEN. PEN. Primary endosperm nucleus is formed. And that process is called triple fusion. What fusion? Triple fusion. This process is called triple fusion. So there are 2 fertilizations are there, 2 fusions are there. Since 2 fusions are involved, angiosperm fertilization is known as double fertilization. Since 2 fusions are involved, angiosperm fertilization is known as double fertilization. What fertilization? Double fertilization. Very important topic. Double fertilization. Double means 2. What are the 2 fusions? Zygamy and triple fusion. What do you mean by zygamy? There are 2 male gametes are discharged. 1 male gamete fused with egg cell and forming zygote, diploid zygote. That is called zygamy. Another male gamete fused with polar nuclei or diploid secondary nucleus and forming triploid primary endosperm nucleus. Triploid primary endosperm nucleus. That is called TPEN or PEN. PEN, primary endosperm nucleus. What is the ploidy? Ploidy means sets of chromosome. How many sets of chromosome? That is called ploidy. So there is a question. You predict the ploidy of zygote. Ploidy of zygote means diploid. 2N. You predict the ploidy of primary endosperm nucleus. Triploid. You predict the ploidy of gametes. Gametes are always haploid. So ploidy. Ploidy means how many sets of chromosome. Gametes are always haploid. Zygote are always diploid. So you predict the ploidy of zygote means that is diploid 2n. Then primary endosperm nucleus triploid 3n. Gametes haploid. So ploidy means how many sets of chromosome. Since two fusions are involved, angiosperm fertilization is known as double fertilization. It is an event unique to angiosperm. This event unique to angiosperm. This fertilization is known as double fertilization. What are the two fusions? Syngamy and triple fusion. What do you mean by syngamy? Haploid male gamete and egg cell fused to form diploid zygote. That is called syngamy. Then what do you mean by triple fusion? Remaining male gamete fused with the polar nuclei or diploid secondary nucleus and forming primary endosperm nucleus. Triploid primary endosperm nucleus. That is called a triple fusion. Since two fusions are involved, since syngamy and triple fusion, two fusions are involved, angiosperm fertilization is known as double fertilization. It is the event unique to angiosperm. It is only found in or seen in angiosperm. In the case of human beings, there is only one fertilization. Our sperm and ovum or egg fuse and forming zygote. Zygote developed into embryo and embryo developed into fetus. Okay. In the case of angiosperm, there is two fusions are there. One is syngamy and another is triple fusion. Since two fusions are involved, angiosperm fertilization is known as double fertilization. Double fertilization. Very, very important. Then what happened to zygote after fertilization? After fertilization, the zygote developed into embryo. After fertilization, the zygote developed into embryo. Development of embryo from zygote is called embryogenesis. Okay, we already discussed in first chapter, reproduction in organism. Formation of embryo from zygote is called embryogenesis. Then after fertilization, zygote developed into embryo. Diploid zygote developed into diploid embryo. Then what happened to primary endosperm nucleus? Primary endosperm nucleus developed into endosperm. Primary endosperm nucleus developed into endosperm. So what happened to zygote and endosperm after fertilization? After fertilization, zygote developed into embryo and a primary endosperm nucleus developed into endosperm. Endosperm also triploid in nature. Okay, after fertilization, zygote developed into embryo. After fertilization, diploid zygote developed into diploid embryo. Then what happened to pen? Primary endosperm nucleus developed into endosperm. Okay, both are triploid. This is about double fertilization. Double fertilization. So we complete double fertilization, pollen pistil interaction, artificial hybridization, outbreeding devices, then agents of pollination. Abiotic agents, biotic agents. In abiotic agents, there are two pollinations are there. What are the two pollination? Wind pollination and water pollination. Wind pollination is called anemophily and water pollination is called hydrophily. And we discussed the characters of anemophilous flowers and hydrophilous flowers. Very, very important. And examples. Okay, what are the examples for wind pollinated flowers or anemophilous flowers? Coconut, grasses, cannabis, date palm. Okay, these are examples for wind pollinated or anemophilous flowers. Then what are the examples for water pollinated or hydrophilous flowers? Vallisneria, Zostera, Hydrilla. These are examples for Ceratophyllum. These are examples for water pollinated flowers. In majority of the aquatic plants like water hyacinth and water lily, flowers are emerged above the level of water and pollination occurs with the help of wind or insects. Okay. Example, water lily and water hyacinth. Water hyacinth is called terror of Bengal. It is a most problematic aquatic weed. It quickly spread over the water body and consume oxygen. It will reduce the or depleting the dissolved oxygen that affect the survival of other aquatic organism. That's why it is known as terror of Bengal. Quickly spread over the water. Okay. Ready? Then we complete about biotic pollination. Biotic pollination. It is called animal pollination. Animal pollination generally called zoophily. Beetles, wasps, bees, and birds. These are examples for animal pollination. Wind pollination is called anemophily. Water pollination is called hydrophily. In the case of biotic pollination, insects pollinations are most dominant. Insects are the most dominant agent. Among the insects, bees are the most dominant agent. Insects pollination generally called entomophily. Entomophily. Then bird pollinations are called ornithophily. Bat pollinations are called chiropterophily. Chiropterophily. Then snails pollinations are called malacophily. Snail pollinations are called malacophily. Then myrmecophily. Ant pollinations are called myrmecophily. Then entomophilous flowers. Connection between yucca and moth. Yucca and moth cannot complete their life cycle without each other. Okay. Then characters of entomophilous flowers. Floral rewards. Floral rewards like pollen grains and nectar. Okay. Then characters completed. So there are three characters. Characters of anemophilous flowers, characters of hydrophilous flowers, and characters of entomophilous flowers. These three are very important. Characters of anemophilous, hydrophilous, and entomophilous flowers. Then we complete pollen robbers, then artificial hybridization, pollen pistil interaction. Now double fertilization. So there are three events: pre-fertilization events, fertilization events, and post-fertilization events. Now we complete about pre-fertilization events and fertilization events. Then third and last events are post-fertilization events. Post-fertilization events. Next topic is post-fertilization event. Our next topic is post-fertilization event. Third event, post-fertilization events. Okay, so we completed about pre-fertilization events, fertilization event, and post-fertilization event. Within the pre-fertilization, we discuss about gametogenesis and gamete transfer. Gametogenesis means formation of male and female gamete. For that, we discuss about structure of male reproductive system and female reproductive system. Male reproductive system means andrecium. Female reproductive part is gynecium. Andrecium, among the andrecium or within the andrecium, we discuss about stamens, anther filament connective, structure of anther, anther wall, epidermis, endothecium, medulla, and tapetum, structure of pollen grains, pollen products, microsporogenesis. In the case of gynecium, we discuss about structure of ovule, stigma style ovary, within the ovary, ovule present, structure of ovule. Okay, structure of pollen grain, then megasporogenesis, and we compare with megasporogenesis with microsporogenesis. There is a table. Okay, microsporogenesis and megasporogenesis. Formation of megaspores from megaspore mother cell within the megasporangia is called megasporogenesis, and formation of microspores within the microsporangia is called microsporogenesis. That's about pre-fertilization. Then pollination, that means gamete transfer and gametogenesis are within the pre-fertilization. Pollination is called gamete transfer. Pollinating agent, autogamy, geitonogamy, synogamy, all completed. Next topic is post-fertilization event. Post-fertilization means after fertilization. Pre-fertilization means before fertilization. Fertilization events means during fertilization process. Okay, so next our topic is post-fertilization event. So what are the main post-fertilization event? We know after fertilization, zygote developed into embryo. Zygote developed into embryo. Okay, then primary endosperm nucleus developed into endosperm. Endosperm is a nutritive tissue that provides nourishment to developing embryo. When zygote developed into embryo, the nourishment provided by endosperm. Okay. So diploid zygote developed into diploid embryo. Triploid primary endosperm nucleus developed into triploid endosperm. That endosperm provides nourishment to the developing embryo. Development of embryo from zygote is called embryogenesis. During embryogenesis, endosperm provide nourishment to the developing embryo. Okay, then what happened to the next? Sepals, petals are fall off. Then synergids, antipodals are degenerate. Then what happened to ovule? Ovule developed into seed. Ovule developed into seed, and ovary developed into fruit. Okay, then covering of ovule is called integument. Integument developed into seed coat. Ovule developed into seed, then covering of ovule developed into seed coat. Seed coat has two layers, outer testa and inner tegmen. Seed coat has two layers, outer testa and inner tegmen. Seed coat has two layers, outer testa and inner tegmen. So integument developed into seed coat. Seed coat has two layers, testa and tegmen. Outer integument developed into testa. Inner integument developed into tegmen. There are two layers, integument, outer integument and inner integument. We know integument developed into seed coat. Seed coat has two layers, outer testa and inner tegmen. Outer integument developed into outer testa. Inner integument developed into inner tegmen. Seed coat has two layers, outer testa and inner tegmen. Okay, ready. Ovule developed into seed, ovary developed into fruit. These are post-fertilization event. Post-fertilization event. What are the post-fertilization event? Post-fertilization means after fertilization. Zygote developed into embryo. Primary endosperm nucleus developed into endosperm. Sepals, petals are fall off. Synergids, antipodals are degenerate. Ovule developed into seed. Ovary developed into fruit. Ovary wall developed into fruit wall. Fruit wall is called pericarp. Fruit wall is called pericarp. Ovary developed into fruit, then ovary wall developed into fruit wall, and that fruit wall is called pericarp. Then integument developed into seed coat. Seed coat has two layers, outer testa and inner tegmen. Outer integument developed into outer testa. Inner integument developed into inner tegmen. These are called post-fertilization events. Post-fertilization events. Okay.